Okay, I think we'll get started. There was one comment in the Slack um, while we were on break, and I've added actually a couple slides for that, and happy to talk about it in more detail. Did anybody else have any topics in particular? You can just unmute yourself if you want, that you really wanted more detail on or a deeper explanation of.
Hey, yeah, this is Margot. I was curious to hear more about the idea of like redundancy in the immune system, and I guess kind of curious like how you think about redundancy when like designing a model, which I guess might come a little later. Yeah.
Excellent point. Any other questions or areas?
Hey, so my name is Devin, and I'm just going to follow up on what Margot was saying. I'm interested in understanding how to include more immunology related data in a hypothesis testing scenario when it comes to modeling. So if that'd be covered, that'd be great.
Sure. Okay. As if you think of something else, put it in the chat, put it in Slack, or raise your hand. I'm gonna, let me pull up participants. Okay, I think I've got all the windows open now, and we will get underway. So I'll go quickly through this, through slides. As I said, it's largely the presentation that's online. I've messed with it a bit, and I'll pause to spend some more time on some of these topics that have just come up, and then we'll see how far we get. And then as I'm talking, if there's something that pops to your mind that you recall now or think of as wanting more depth on, then let's just talk about it. So one of the first things that you have to deal with in thinking about how the immune system works as an effector mechanism is the fact that it's targeting organisms that are on vastly different scales. And so you have viruses that are quite small, many times smaller than a cell, and function and survive by infecting and replicating within a host cell. And so the mechanism of removal for a virus is usually the killing of the host cell, versus bacteria, which are somewhat larger, usually still much smaller than a host cell, but can either be extracellular or intracellular. So can have both lifestyles. Even the same species of bacteria can have both lifestyles. And so now the host immune system, if it's dealing with an extracellular bacteria, cannot use things as effector mechanisms that would involve or rely entirely on killing the infected cell, and vice versa. And then parasites are usually many times larger than a host cell because they are multicellular themselves, and quite complicated. And so here, as effector mechanisms, a lot of the arms of the immune system that require some cell infection or some fragment of the pathogen to be processed and presented by a cell are going to be largely ineffective. And in thinking about how these pathogens have to be removed from the body, it's mechanically actually a much more difficult problem, because you're actually asking the system to sort of move an object that's many times larger than the average sized effector, which is either a cell or a protein, even smaller than a cell. And so these different scales of size also relate to different scales of pathogen life cycles. And so for viruses, you usually have relatively rapid replication cycles. So you have a cell that gets infected and produces progeny relatively quickly. There are viruses that can live with you for a very long time, but their actual replication cycle is still usually quite fast. You have bacteria and parasites that can live with you usually on a slightly longer time scale, but still are trying to replicate relatively quickly and get out of the host and spread to the next host. And then you have the really complex parasites again, both these are worms, but this applies to malaria as well, where they are looking for really, a multi-year lifestyle in their definitive host and are manipulating the immune system to achieve that goal. And so the immune system has to assess the variation between these factors because it will often respond relatively robustly and overwhelmingly to an infection with something that can kill the host quickly. And so that's going to be things like bacteria and viruses. And that's the result of evolution driving effector mechanisms and sensing mechanisms that realize the scale of the threat. And it will respond often much more slowly and methodically to an infection that is less of an immediate threat and requires maybe a more sophisticated response. And so the questions that the immune system has to answer are, is what I'm actually facing a threat? Is it infectious? Is it not self? And even if it's not self, is it infectious not self? And is it pathogenic not self? And we have a few people that introduce themselves saying they're working on microbiome. And this is one of the key questions in trying to understand the relationship between the host immune system and the microbiome is how the immune system distinguishes, beneficial microbiome from pathogenic microbiome, and the fact that certain organisms can actually be both depending on what tissue they're in and what state of their life cycle they're in and sort of metabolically how they're what they produce and how they're regulated. And then even if the immune system decides to respond, it has to decide what effector mechanisms are going to be employed. So what are going to be the most effective means of control, and how does tissue damage get limited, and how does tissue repair occur in a rapid enough fashion that the organ that's infected restores itself. And so these are sort of some general immunological themes that I think are useful for keeping in mind, even if you lose a lot of the details. If you develop sort of a gut sense of how the immune system works, it can help you out a lot. I think even in your modeling of thinking about what's likely important and what the general principles are of an immune response. And so almost every infection, regardless of how it starts and regardless of how it ends, initiates with this generalized inflammation followed by a specific pathogen-directed response. So the host responds with a more generic response initially that becomes more and more specific as time goes on. And that's basically reflective of the information flow into the immune system. So the earliest events in immune recognition are usually much less specific than the later events, and the system itself is capturing more and more information as time goes on to be able to direct the response in a more specific fashion. The second principle is that there's a lot of communication via soluble mediators. And so you'll see these words a lot, cytokines and chemokines, and you'll see these measurements a lot, particularly those of you that work in any kind of human system. These are very easy things for us to measure, and we can measure a lot of them at one time. And so we like to measure them because they're easy to measure, it's relatively cost effective, and we can measure them all at once. And so modeling the cytokine and chemokine network can be, I think, incredibly fruitful in terms of getting a snapshot of the state of the immune system at any given moment. But you do have to keep in mind that the soluble mediators are at different concentration gradients in different tissues. And the tissue that we often have the most access to in the human is in blood, which may not be reflective of the actual site of inflammation for something like COVID. There's a lot of feedback regulation to limit hyperinflammatory responses. And again, I list this here because I think it's something that should figure into a lot of models, in that every sort of action by the immune system usually triggers some kind of negative feedback loop that will dampen the forward signal. And this can be read out at the level of cytokines, it can be read out at the level of gene expression, and it can be actually read out at the level of particular cell populations that get recruited to the response. And then the point that was just raised about redundancy, there's multiple modes of recognition and specificity determination. And so when you're thinking about a pathogen, the best way of dealing with that is trying to figure out if there's data that allows you to distinguish between these different modes of recognition and specificity, and which ones have which specific effects. Where is their redundancy? Where is their specificity in the actual responsiveness? And can I capture that in the model? And or can I collapse things in the model that I don't really need to have as separate elements? This comes back also to when we measure 50 different cytokines and chemokines, a lot of times 20 or 30 of them even will be highly, highly correlated with each other. You know, R squared values above 0.9 for a whole group of cytokines and chemokines. And while I could tell you how they're all very different from each other and why this one signals through this pathway and this one signals through that pathway, if you really just want sort of the net effect of that module of chemokines in your model, you maybe just have to have it as a single factor, and it can vastly reduce the complexity of your model. It also can vastly improve the power of any type of statistical analysis you're doing if instead of testing 20 or 30 different cytokines, you actually just test the model and do some sort of feature selection. A really critical idea that I think actually is hard for a lot of immunologists to wrap their heads around is that the immune system is not centrally organized. And so while we often talk about teleology with the immune system and what it's trying to accomplish, it is the evolutionary consequence of a number of different independent systems, many of which evolved out of other, sort of other purpose tissues. So, functional tissues that were doing something else in the organism that started adopting a role in immune responses. And so while they're obviously all integrated with each other, it's not that there's an individual a locus of decision-making that's picking which responses are going to occur, even if I talk like that a lot. And even still, even in our most studied infections, it's not always clear what is essential, what is redundant, and what is irrelevant. We can look at this in animal models, but no animal model is perfect, just like no mathematical model is perfect. And you have to use some amount of your own judgment to try to figure out both what the data suggests are these key features, what's robust in the data, and then how you might actually formally test it with data that's available to you. Okay. So thinking from the pathogen side and thinking about the pathogen-specific approaches that the immune response uses, the evolving response has to really start getting specific in terms of eliminating a particular pathogenic threat. And so some pathogens must be eliminated for host survival. These are usually small, rapidly growing pathogens with limited genomes. And here the pathogen is really not caring as an evolutionary unit. It doesn't really care much about the individual. The primary goal for the pathogen is to win a race, get in and get out as quickly as possible. And so the best example of this is something like influenza, Ebola, RSV, SARS-CoV-2, where the infection is going to produce billions to trillions potentially of viruses, and really only a couple hundred of them have to get out of the host and into a subsequent host for that infection to have been successful. And there's relatively low cost for the pathogen to make a copy of itself. Often this isn't particularly sophisticated in terms of the way the pathogen functions. You'll see there's some level of evasion that occurs, and the larger the genome of the pathogen, usually the better the evasion mechanisms. But there's not anything approaching some of the clever tricks that larger pathogens use. Some pathogens are acute or chronic depending on the host situation, and if a pathogen can achieve chronicity, they're usually more complicated. The decision of whether or not it becomes an acute or chronic pathogen is usually some interaction between the host and the pathogen itself. So host genetic and environmental factors might determine susceptibility and permissiveness. And examples here would be something like TB, which people can eliminate on a primary infection if they mount a robust enough immune response. Viruses like HCV or HIV, which are not super large, but have very clever mechanisms for either ongoing evading the immune response or integrating into the host genome in a way that allows them to persist. And then you get into the larger pathogens that are almost exclusively chronic. And I include here the large viruses like the herpes viruses that have hundreds of genes that are targeted towards manipulating the host and maintaining chronic infection without integrating into the host genome. And many times we still don't fully understand in every circumstance how some of these pathogens maintain a chronic lifestyle. And it may be actually failures of our own immune systems that support this. So if the immune system just can't come up with an effective mechanism, then by default, the infection can become chronic. These are usually not acutely lethal. So they're sophisticated enough to maintain the relative health of the host such that they're provided with their metabolic and environmental needs. And the interaction of host metabolism with pathogen metabolism is something that's becoming, I think, much more a focus of research, particularly as metabolism is just becoming such a central focus of cell biology research, both for immunology and for other arms of the response system. And that's a good way of sort of getting a handle on what the pathogen wants versus what the host is trying to block. And the examples here would be something like parasitic worms, particularly in terms of the large pathogens. And so you can think of this, this sort of scale of responsiveness where the initial response is really going to be to try to just block colonization by the pathogen by mechanophysical blocking. And if that fails, there's the early innate response, which is going to involve relatively nonspecific, though not completely nonspecific inflammation and effector cells that will try to limit the course of the infection. If you do not have this response and due to some sort of genetic dysfunction, you're a highly susceptible individual that is often, has a very difficult time sort of thriving, regardless of what species you are. If this fails and the pathogen mounts a reasonable rate of replication, then you start developing classic adaptive immunity, and what we often think of when we think of the immune system, which is the part of the immune system that is memory, so B cells and T cells. But if this has to be mounted, you're sick. You probably don't feel well, and you're going to remember this as having a cold up through having a more severe bout of illness. And so most of the pathogens that we encounter are stopped here at these two levels. And the failure of these two levels, again, are really much more catastrophic in a way than the failure of either the B cell or the T cell response. Some people live their whole lives without B cells now because they can just be treated every so often with polyspecific IgG. I'll come to some of the questions at the end, and then I'll try to incorporate some of these as they come through. And so please feel free to keep putting them in, and then if I don't answer them at the end, we can revisit them. So just recapping that, the stages of the immune response are this physical prevention of infections, so blocking by the skin, mucus, innate epithelial inflammation. So those first layers of the body that a pathogen usually encounters are usually epithelial layers. So your skin is an epithelial layer, your lungs are an epithelial layer, and your gut are an epithelial layer. And these are all the surfaces that have exposure to the external world that are going to be where the pathogens mostly enter. There are some pathogens like dengue that was just mentioned in the chat that will come in directly into the blood, and so there'll be endothelial and blood cell inflammation. And then you have the round of professional innate cell recruitment, which can have direct effector mechanisms that attack the pathogen. And then if all of these fail, you generate your adaptive immune response, which includes CD8, CD4 T cells, and B cells. And so this is kind of it's an old review, but you see this image a lot because it's a nice quick summary of the different cell types that are involved in these different responses. And so you have this fast response that's relatively non-targeted, relatively non-specific, that involve these innate effector cells like macrophages, dendritic cells, and natural killer cells, and then these other granulocytic cells, basophils, eosinophils, and neutrophils. These are sort of some of the less understood cells in the immune system. They're harder to study. They're very fragile. There's fewer people that work on them. These are macrophages and dendritic cells are incredibly well-studied cells in the immune system. And so that's another thing that you have to balance when you're trying to interpret data and model it, is sometimes the reason you have a lot of access to data is because it's something that people have studied a lot, but it doesn't necessarily mean that it's actually the most important thing in the response. In contrast, on the adaptive side, you have the classic B cells and T cells. B cells make antibodies. T cells come in CD4 and CD8 flavors. And then sort of bridging these two, you have these innate lymphocytes, which include gamma delta T cells, natural killer T cells. These are T cells, but they aren't particularly specific. So they respond to more pan type signals that are not characteristic of any individual pathogen. I'm going to skip over these slides just again because this is in the online talk, and move towards spending a little more time on a couple of slides on innate recognition, because that is something that came up in the chat and is more and more something that people are trying to understand, I think, through modeling. And so the initial way that a cell, any cell in your body, whether it's a professional immune cell or a structural cell like an epithelial cell, which would be like your skin cells, your lung cells, your gut cells, recognize that they've been infected is through this process called pattern recognition. And the concept of pattern recognition really emerged in the 80s, but it came about as part of a really decades-long understanding that there was something we were missing in understanding the immune response. The sort of 60s through the 80s was this explosion of understanding about adaptive immunity, and we'll talk a little bit more about that in a few minutes. But the excitement about the immune system was really about immunological memory, vaccination, and the ability of eliciting these B cell and T cell responses against pathogens that would prevent you from ever getting that pathogen again. But all of those vaccines that worked really well had some element to them that was actually not the target of the immune system. And so there were various compounds that were used to sort of juice up the vaccine and make it work better. And collectively, all of these things are called adjuvants. And what adjuvants do, retrospectively now we can say, is they activate innate immune responses and they activate innate immune recognition, which is really critical, this inflammation, to getting the rest of the immune system to turn on. But nobody really understood that. It was sort of accidental. It was, the fact that we actually couldn't make things in a very clean way. And so adjuvants snuck their way into a lot of preparations because they were just natural contaminants of a lot of the processes that were used to make vaccines. And this became sort of codified by Charlie Janeway, who was a great figure in immunological history. He's died now almost 20 years ago. But he called adjuvants the immunologist's dirty little secret, because it was something that mechanistically wasn't understood and but was absolutely necessary to making effective vaccines. And so he really, from a pure theoretical perspective, started thinking about what adjuvants must be doing. And he came up with this concept of pattern recognition, with the idea that what adjuvants were things that mimicked patterns that had to be in pathogens, that were patterns that weren't in non-pathogens, that weren't in us, and that weren't in non-threatening bacteria, non-threatening viruses, and non-threatening other things that we encounter in our lives. And so he, in the late 80s, wrote this transformational essay where he proposed the system that there would be what he called pathogen-associated molecular patterns, which would be things that appear in viruses and bacteria that don't appear in normal, healthy human cells. And there would be receptors for those pathogen-associated molecular patterns. So he called those PAMPs, and he called the receptors pattern recognition receptors. And then he went on to be one of the labs that contributed to the early discovery of some of the first pattern recognition receptors. And then he unfortunately passed away, but he very, very, very likely would have won the Nobel Prize if he had survived, because there was eventually a Nobel Prize for pattern recognition that was won by other people and was hugely controversial because some of the people were not thought to have earned their spot in the list. But The very first pattern recognition receptors that were discovered, including by Charlie Janeway, were the toll-like receptors. And so these are called the TLRs. They're called the toll-like receptors because toll is a protein in Drosophila that they look a lot like. And these are proteins that are either on the surface of cells or inside vesicles in cells. And they bind to exactly what Charlie Janeway predicted they should bind to. They bind to patterns that appear in pathogens that don't appear in host cells. So the classic adjuvant and the classic first ligand of a TLR that was discovered was for TLR4, and that ligand was something called LPS, which is lipopolysaccharide, which is a component of the surface of bacteria. And so this lipo, meaning just fat, polysaccharide, meaning sugar. So this fat-sugar combination is something that never occurs in a healthy human. It never is made by your healthy human cell. And so if you see LPS floating around, it means you've got a bunch of bacteria somewhere that you probably don't want. And so this can be recognized by TLR4, and it starts a signaling cascade inside the cell that leads to this transcription factor activation that then drives inflammation. And the inflammation can occur in the structural cell itself, so in the epithelial cell. So virtually every cell in your body is ultimately an immune cell, and that it can try to deal with the infection itself. It will often fail, but it will mount this response. And in mounting this response, in addition to trying to kill the pathogen itself inside the cell, which it has a relatively limited armamentarium for, it will secrete cytokines and chemokines outside the cell to alert the professional immune system that there is a problem. There subsequently have been discovered two other very large families of pattern recognition receptors, so the NOD-like receptors and the RIG-I-like receptors. The NOD-like receptors are a giant family of receptors. There's some shown here that agglutinate together and activate these enzymes called caspases that actually lead to the direct activation of cytokines and chemokines for secretion, and also can initiate cell death. And so one way that a cell can very effectively deal with an infection is it just kills itself, and it will stop the propagation of the infection. There are about 30 of these in humans, and we have really good functional understanding of maybe 6 of them. There are like 100 of these in plants, and it is the primary way that plants have an immune system. And then the last group is called the RLRs, and these, you don't hear this term as much anymore, but you do still hear it sometimes. It gives, so the RLRs recognize nucleic acids. And nucleic acids are a great way to recognize that there's a problem, because nucleic acids like RNA and DNA have very specific spots in the cell that they should be. And if they're not in those spots, then it usually means that something is not where it's supposed to be. And so if you've got a bunch of DNA in your cytoplasm when it should be in the nucleus, it probably means that you have a virus that's a DNA virus, or a bacteria that has its own DNA. And if you have certain structures of RNA in the cytoplasm that aren't the classical eukaryotic RNA structures that should be in your helping you make proteins, then it also probably means you have a virus that has some weird nucleic acids. So this is the core principle, is pattern recognition. And so going to your question, Jason, about how do you memorize all of these things, you can't, unless you're going to devote yourself to it, it's pretty hard to memorize all of these things. You can have great references where you check and see where the field is. But it's good to just have a general sense that if there's a pathogen, there's probably a pattern associated with that pathogen. What are those patterns? How are they generated? And what's really critical for my model, and how, what part of the life cycle and of the immune response am I actually trying to model? I've added a couple slides here to show you how I like this slide still. A lot of people still use it still because it shows the degeneracy of the signaling of these pathways, that they all often converge on the same NF-kappa B and other transcription factors, leading to some of the same effects. But the pathways have just gotten more and more complicated. More and more pattern recognition receptors are being discovered, and the localization of these pattern recognition receptors is getting more and more refined. So the the isolation of specific pattern recognition receptors, particularly the TLRs within specific spots in the cell, has become a really key element of how they work and how they work effectively and how they don't go rogue and start getting upset about things that they shouldn't get upset about. As I said, we're still working on resolving some of the roles of the NLRs. Probably the biggest change in the last 5 to 6 years in terms of a new area of pattern recognition has been the expansion of the types of nucleic acids that are recognized. And so that's over here on this chart here. All of these are pattern recognition receptors that are part of various other systems. The biggest kind of new one that was a big a big shock to the system when it was discovered was C-gas. So it it recognizes various forms of double-stranded DNA, and then it modifies those forms of DNA to make the second messengers that then trigger downstream immune responses. To give you an example of how active this field still is, this slide is itself even a couple years old, and it has on here this protein dye, which is unfortunately in immunology, there's a lot of double naming of things. It's also called ZBP1, and ZBP1 is the preferred name for this now. But it was only in about 2016 that it was recognized that this protein actually probably plays the critical role in recognizing nucleic acids for influenza virus. And this is after literally 15 to 20 years of people studying the innate immune response to influenza virus and assigning roles to various other molecules, which do play a role. So for influenza, since I know we have some influenza people on there, TLR7 plays a big role because influenza is a single-stranded RNA virus. NLRP3 can play a role, and some of the Regai-like receptors can play a role. So Regai itself actually can recognize influenza. But one of the really dissatisfying things about Regai, NLRP3, and TLR7 as molecules that recognize influenza is that you can knock out all three of them, and the phenotype wasn't really super impressive, meaning that the mouse still kind of made a pretty good immune response to influenza. And so it wasn't until this discovery was made that it was really understood that there was probably yet another pathway that was potentially even more important than the others, driving the influenza response. And as you'll see on this slide, they said here that this was a double-stranded DNA response. And what has been discovered literally in like the last year and a half is that it's actually not double-stranded DNA. It's Z-form RNA, which is a very specific twisting form of RNA itself. You can also get Z-form DNA, but the actual existence of Z-form RNA was actually still controversial even in the last couple of years. And so the field's still moving, very quickly, and really fundamental discoveries are still being made, which is, potential complexity for trying to model anything with high resolution. Okay, and then I'm going to go quickly now through some of the other innate slides, and then just so that we have some time to talk about adaptive immunity, and then I'm happy to spend time on specifics for anything else. So once the recognition occurs, you start getting these degenerate pathways, and by degenerate I mean that all of these pathways start converging back on conserved effector mechanisms that can occur in virtually any cell type in the body. And that includes the interferon response, which you'll hear a lot about, particularly in the context of viruses, but you also hear about it in bacterial infections and even in some parasitic infections. And interferons were some of the first cytokines discovered. They were named sort of intuitively in that they interfere with viral replication. But they themselves are cytokines. So they're not actually doing much themselves. They're a signal. They're a signal to surrounding cells that there's some kind of infection. And they're actually an autocrine signal back on the cell that's making them. So they'll feed forward and amplify this idea that the cell now has that there's an infection. And what they'll do is they'll turn on a whole host of genes that are not normally on in a cell that will try to actually block replication of the pathogen. And particularly for viruses, this will take the form of degrading any stray nucleic acid inside the cell. And so it induces a whole bunch of enzymes that will just go around and start chewing up RNA, and it will chew up a lot of host RNA. And so cells that are inducing a strong interferon response will actually start losing the ability to make other proteins inside that cell. So it's a stressed response. It's not something you want to be in all the time, but it's doing that to try to block the effective replication of the virus. We'll also do things, interferons will do things to help recognition by more professional cells within the immune system. On the bacterial side of things, for things like TB, or on the parasitic side of things, for things like malaria, you have these professional cells of the immune system, macrophages and dendritic cells, that can come along and will start eating cell debris in the area and any pathogens that are floating around in the area. And so they will literally eat and take in the pathogens, and they will put them in little vesicles inside the cell, and they will try to kill the pathogen inside those vesicles. And they'll do that through chemical means. And so the innate activation that's occurring on the epithelial surface that's causing the secretion of these cytokines will license these cells to start making toxic chemicals like nitric oxides and reactive oxygen intermediates that they will try to keep contained inside a vesicle inside the cell, that they'll fuse with any vesicle that has pathogen in it, and it will chemically destroy the pathogen. Again, these are highly inflammatory chemicals, and they're going to leak out of the cell at some level, and they're going to cause damage to your surrounding cells. And so when you watch commercials for vitamins that they say you should take antioxidants, the antioxidant is the is a scavenger to actually absorb these reactive oxygen intermediates and reactive nitrogen intermediates, which you are generating all the time because you're always faced with pathogens of some form or another. And as this fails to work and the pathogen continues to replicate, you will have further amplification of the immune response. So we had signals coming from the initial surface of infection, the epithelial cell, and then we had some professional macrophages wander by and try to deal with the infection. And if they failed, they're going to start amplifying this inflammation even further by secreting more cytokines themselves to alert more of the professional immune system to come to the site of infection. So more macrophages, more dendritic cells, neutrophils, eosinophils, all those granulocytes, to start trying to sop up the pathogen and kill the cells that are infected with the pathogen. And they're going to be setting up the possibility of an adaptive immune response. And so all of this inflammation is necessary to get the adaptive immune response interested in responding. And so you have these classic early inflammatory cytokines. The three that you'll hear the most about and that are often measured are IL-1 beta, TNF, and IL-6. These all have direct effects on cells to try to, again, turn on some of those effector mechanisms to try to get them to either kill themselves or make those enzymes to kill the pathogen directly inside the cell. And then they're also going to start mobilizing the adaptive immune response itself. But these are once you start making these cytokines and chemokines is when you start feeling bad. These cause fever. These actually, if they're produced in too much concentration, will cause shock. And this can actually be a deadly immune response if it gets out of control. This is what everybody felt bad with probably after their COVID shot, particularly after their second COVID shot, if you got one of the RNA shots, is this amplification of inflammation that caused fever, aches and pains. It's all your host immune system signaling that there's some inflammatory response that needs to be dealt with. Part of what these cytokines start licensing is changes in your endothelial layer. So the endothelium are the cells that line your blood vessels. And when they have receptors for all of these cytokines and chemokines on them, and when they get a signal that there's local inflammation, so let's say there's an infection in your tissue, we can pretend this is your lung, and your lung cells are saying we need cells out here to clean up these viruses or these bacteria. And the epithelial cells and the local macrophages have started making cytokines. They'll tell this endothelial layer that they should start allowing cells to pass through. And so the endothelial layer, you've got your bloodstream right here, cells are flying by. It's starting to upregulate these adhesion molecules to say, we need cells on the other side of this blood vessel, and you should stick to me, and I'm going to let you through in between two cells. And they will squish themselves through this very tiny space in between these two cells, and you'll get cells into your tissue that will start helping deal with the infection. This process, though, is not perfect either. And so you've got all your blood flow over here, you've got this hole opening up and to let the cell through. And even though it's a very tight fit, you're going to have fluid that leaks through into the tissue. And this is why you get all kinds of weird swelling and redness when you have an infection. If you cut yourself in your skin, this is all the redness that you get around the site of the cut. It's all that liquid leaking through along with the cells to come deal with the infection. The last thing I'm going to talk about for the innate response is complement activation. And this is a really good example of both redundancy, convergent evolution, and the fact that the immune system is not centrally organized. And so complement is this very ancient form of innate recognition. You can even think of it as a form of pattern recognition that's unrelated to any of the other receptors that we've been talking about. And it is this set of proteins that is constantly circulating in your blood, and that when activated can be an incredibly potent source of pathogen killing. But it sort of exists in this parallel track to what all the other things that we've been talking about. There are some points of interaction, but there's not a ton of overlap. And complement's really important because people with defects in complement show up with all kinds of interesting infections. And so there are multiple ways to activate the complement path cascade. And this is one of the things that people hate learning the most in immunology. And so I'm not going to dwell too much on the details. But the complement cascade, as I said, it's the set of proteins that we, as immunologists, tried to do a good job of naming, and that we tried to name them all just C1, C2, C3, C4, C5, C6, C7, and so forth, which is the cascade. But they were discovered out of order. So the actual activation of the complement cascade does not occur in the numerical order. And so you have to memorize this when you're an immunology student, and everybody hates it because it's very trivial. But the key thing to keep in mind is that you have this cascade of the initial recognition of a pathogen by the early parts of the complement cascade. So C1, luckily, is one of the first ones that forms this recognition, and it forms it through pattern recognition. So it sticks to certain structures on the surface of bacteria, and primarily bacteria. And if it sticks enough, it recruits the next element of the complement cascade. And if that sticks enough, it recruits the next element of the complement cascade. And if they stick enough together, they start forming enzymatic active sites that self-cleave themselves and start this runaway train of further complement activation until you get this complex formation. So now we're up to C5, 6, and 7 on the surface of the target that the complement initially recognized. So often that's bacteria. It can also be an infected cell that is expressing some bacterial or viral protein. And these complement proteins will start inserting themselves into the membrane of either the bacteria or the host cell, and then recruiting large amounts of this one complement component called C9 that form this rosette inside, binds to itself and forms this rosette inside the membrane, and is basically forming a pore in the membrane. And when it finishes, it forms this perfectly circular pore. So this is just a circle of C9 molecules. And this happens fast. This happens in under a second once it starts happening. And it happens thousands of times over the surface of the target, whether it's bacteria or a host cell. And so you go from a totally intact membrane to a membrane that's now shot through with hundreds to thousands of these holes formed by the C9 complex. And the C9 is circulating in your body all the time. And so the cell basically just explodes, or the bacteria basically just explodes. So it's incredibly potent. again, this is sort of this parallel response that relies on complement itself to do the initial recognition. Complement has now evolutionarily adopted being able to be activated by antibodies itself. So an arm of the adaptive immune response that we're going to talk about here in 5 minutes or so, it can recognize antibodies sticking to something and will jump on the antibody train and try to kill whatever the antibody's bound to. And these pores that it forms look almost exactly like the pores that are formed when T cells kill a target cell, but it evolved independently. So it looks very similar, but it's evolutionarily, the gene products are unrelated. It's the mechanism itself is basically just the same mechanism. Okay, I'm gonna skip now to adaptive immunity, and to bridge into adaptive immunity, I just want to mention that another relatively recent evolution in, evolution is a bad word here, discovery in the immune system is the concept of innate lymphocytes. And so we're going to talk about adaptive immunity. Adaptive immunity is mediated by lymphocytes. Lymphocytes are B cells and T cells generally, but there are these innate lymphocytes that basically look like T cells without a T cell receptor. And we're going to talk about the importance of the T cell receptor for mediating specificity. But innate lymphocytes are these cells that have all the functions of a T cell, but no T cell receptor. And I think this is, we're sort of, I think, in the middle of a complete re-understanding of adaptive immunity and the evolution of lymphocytes and what they're really for, because we started with the discovery of lymphocytes from this perspective of cells with these specific receptors that form memory, but there's really a much more ancient immune response underlying underlying that system that evolved long before there was any T cell receptor or B cell receptor or antibody. And so these cells play a really key role in amplifying inflammatory signals and in tissue repair and in tissue damage. You're going to come across them, but they're sort of still a little bit mysterious, I think, in terms of all of their functions and how they bridge innate and adaptive immunity. Okay, so I'm going to switch here to adaptive immunity and spend just about 10 minutes on B cells and T cells really quickly, and then talk about some of the special topics that were raised. But there are three broad arms of adaptive immunity, and this is, again, classically what we're talking about when we talk a lot about the immune system and when we talk about vaccines and interventions. So you've got B cells that make antibodies. The antibodies are soluble effector molecules that participate in extracellular pathogen removal. I call them little torpedoes of the immune system. They can be made in high quantity and flood your blood, can flood mucosal surfaces, and literally block mechanically a pathogen from doing what it wants to do. And then you have two types of adaptive specific T cells, CD4 T cells and CD8 T cells. CD4 T cells are really highly regulatory, so they induce other host responses. They are, if you were going to put a cell at the center of an immune response, in a fully formed immune response, you could argue that the CD4 T cell is making most of the decisions. They are interpreting a lot of signals from soluble mediators and from other tissue factors, and then they're giving a lot of signals in the form of soluble mediators and surface proteins that direct CD8 T cells and B cells, so often called the quarterbacks of the immune system. And then CD8 T cells are often called killer T cells or cytotoxic T cells. They're direct effectors that go out and kill an infected cell. So this is going to be important for various viruses, almost any virus, and some intracellular bacteria and some intracellular parasites. And all three have memory. And I want to just skip ahead to the fact that just like pattern recognition, the concept of adaptive immunity also came from a pure sort of theory based on observation, but a theory that was proposed long before it was ever proven. And so this is the clonal selection hypothesis, which was proposed by McFarlane Burnett, an Australian, who, based on the observation of immunological memory, came up with this idea of how immunological memory had to work. And again, this is a nice kind of way of thinking about the general principle of adaptive immunity without having to memorize and learn all of the nomenclature around adaptive immunity. And so again, if you're building a model and you want an adaptive immune component, if you keep these principles in mind, you can figure out what actual specific cell effector fits this particular model. So the idea of the clonal selection hypothesis was to have memory, and to have memory that was so specific for a pathogen where you would not get exactly that pathogen again, but you might get a very similar pathogen again. McFarland and Burnett came up with this idea that each lymphocyte bears a single type of receptor that has a unique specificity. So you've got a cell that has a specific target, and somehow an interaction between the pathogen, the foreign molecule, and that receptor activates that cell. And so even the concept of cell activation was quite novel. And then those activated cells replicate, and each daughter cell has exactly the same receptor. And then cells that would have a receptor that would target a self molecule have to be deleted in some way. And so he came up with all of these ideas actually before we even knew there were B cells and T cells, and all of these are true. And so all of these have been borne out both for B cells and T cells in different ways. And so for B cells, you have exactly this concept where you have a B cell that actually has a lymphocyte receptor that is the antibody that's actually surface bound initially. So an resting B cell that's never been activated before has an antibody on its surface. It looks like this. We always draw antibodies as Ys. And the antibody can directly bind its antigen. If there's no mediation, it just binds onto its antigen, and it will activate the B cell. And if the B cell gets some instruction from the CD4 T cell, it will start proliferating, and it will start making many, many more of those antibodies. So the really tricky part of this, in terms of it working, is that how is it possible that we can make cells with our relatively finite genome that only has 30,000 genes? How can we make cells that have an antibody for things that we've never ever seen before, and that basically we never have a hole in our immune repertoires? And so the answer to that came sort of in the late 70s, where the discovery was made that if you looked at this is very primitive sort of molecular biology using southern blots, which don't have to go into the details of, but they were ways of looking at the organization of DNA by just looking at size distribution of fragments made through cutting of the DNA. And the thing was that if you looked across humans, different humans, for most genes, these size fragments were sort of the same, would show up the same on these southern blots. And what the first time that so between humans, that was often the case. And within humans, it was definitely the case if you looked across different cell types that all of their DNA was sort of organized in the same way. And the only exception that's really been found to that is in B cells and T cells, where if you look at the B cell receptor or the T cell receptor, the B cell receptor is the antibody. If you look from different B cells to different B cells, the organization of those segments the organization of their DNA is different. And what that's led to is the understanding that in an antibody, which is depicted here, you have these regions of the antibody that are hypervariable. So they are, they mediate all of the antigen specificity, and they are very different between different B cells. And the way that they're generated is through this process called VDJ recombination. And so I'm just going to skip to what VDJ recombination is. And it is this amazing process by which, when you look at the actual genes that encode for antibodies or T cell receptors, it's exactly the same principle. And so we don't have to dwell on it too long for T cell receptors if you understand it for antibodies, is that you have these loci of cassettes of genes. And so these genes are labeled as, or these gene segments are labeled as variable regions, so V, diversity regions, D, and joining regions, J. And they're just lined up next to each other with little tiny introns in between them. And when you are a developing B cell or a developing T cell, and you want to make your B cell receptor your antibody or your T cell receptor, you have a very complicated process by which one of these V regions one of these D regions and one of these J regions is selected and fused together to make your unique antibody or T cell receptor as a cell. And so what that looks like is that the DNA is actually physically cut. I think I have this. Do I? No, okay. Sorry, I want to go back to this. The DNA is actually physically cut, and your V and J or your V, D, and J are stitched together to make the in-frame complete antibody response, antibody for that cell. And so you might have, in a developing cell, you might have 40 different variable regions to choose from, and you're going to pick one. You might have 25 D regions to choose from, and you're going to pick one. And then you have 6 J regions to choose from, and you're going to pick one. And the enzymes are going to cut out all the DNA in between these gene segments that you're not using, and then stitch together the ones that you are using. So everything in between is cut out, and then you stitch them together. And this process is purposefully error prone with DNA getting inserted and deleted somewhat randomly so that you actually generate more diversity. And the diversity can reach really astronomical levels. This is a 2005 textbook. This is still the number that you'll see reported in the Janeway textbook. But the actual number of potential T cell receptors is estimated to be about 10 to the 61. And also something like 10 to the, I think it's 50 something for BCRs. And so this is just based on sort of bad math, but it stays in the textbook. The reason why there's not a huge rush to correct these in the textbook is it's still way more, even 10 to the 18th is way more than the number of T cells you'll ever make in your lifetime. And so the possible repertoire of the T cell receptor repertoire or the B cell receptor repertoire is much vaster than the actual repertoire. So you're subselecting a very small number of the potential TCRs and BCRs that you can make, but that subselection still covers basically any antigen that we come across. And so, so that is how you generate this vast diversity of potential antigen recognition. And in the last couple of minutes here, I'll just mention that. So the key thing to think about antibodies is you've generated this vast potential diversity. So now you can see SARS-CoV-2, even though we've never been infected with SARS-CoV-2 in our entire lives. And the antibodies are directly binding the antigen. The difference with T cell receptors, I'm going to skip to T cell receptors now, is that T cell receptors are never secreted from the cell. So the antibody molecule itself can become the effector molecule. So it can get released from the cell and go out and bind the virus and block the virus from infecting another cell. The T cell receptor is purely a recognition molecule. It's not an effector molecule. And so it's going to bind its antigen on a target cell, and it's going to bind its antigen not as a fully formed antigen, not as part of the virus. It's going to bind just a small peptide from the protein of the virus or the bacteria or the parasite. And so you have your professional immune cells, like your antigen presenting cells, like macrophages and dendritic cells, that are eating these pathogens as they go through a site of inflammation, and they're chopping up peptides from the proteins inside the viruses. And then they're presenting them, and that's the word that we use, is it's antigen presentation on their cell surface, and these special molecules called MHC, major histocompatibility complex. Just to complicate things, when the MHC is a human MHC, we call it an HLA for human leukocyte antigen. And so these molecules just literally push a little peptide out on the cell surface, and they're holding it out there for a T cell receptor to look at. That's the purpose of that system. And the T cell receptor will come along and look at that peptide. It will also look at the MHC molecule. So it sees them both at the same time, and that helps stabilize it. And if it sees the right combination, that's what activates the T cell. There's some discussion in the chat about what CD4 and CD8 are doing. So they're actually participating in this interaction. So CD4 binds to a particular type of MHC molecule called MHC class 2. And then MHC class 2 molecules are only restricted on professional antigen presenting cells. So your macrophages and your dendritic cells that are called in at the beginning of the response, and they have a lot of specialized functions for antigen presentation. and for innate recognition. So they have more TLRs, NLRs, RLRs than any other cell type. So they're really the core fact finders of the innate response. And they're going to tell the organizers of the adaptive immune response, the CD4 T cells, that there's a problem. But they're going to sort of restrict that job to themselves. And so that's teleologically why you can think of why CD4 sees MHC class 2, and all of that's restricted to professional innate immune cells. CD8 T cells see MHC class 1 molecules. There are some differences between class 1 and class 2 molecules that are sort of outside the scope of our course here. But this is sort of what they look like. Again, you're sort of looking straight down on it here. This is the antigen presenting molecule in white, and then the peptide itself in pink here in the middle. So it's like a little cleft, and the T cell receptor is going to sit down right on top of that and visualize it. So CD8 T cells see MHC class 1 molecules, and MHC class 1 molecules are on every cell in your body. And the reason you want that is because your CD8 T cell is your killer cell, and the CD8 cell needs to kill any infected cell in your body. So it doesn't want to be restricted to just the professionals. If you've got an infection in your lung, it's going to go kill your lung cells. If you've got an infection in your leg, it's going to go kill some muscle cells in your leg. And so it's unrestricted, but CD8 T cells have a lot less decision-making power than CD4 T cells, in part because they can be activated by any cell in your body. So I've gone a little bit over because I got chatty from some of the discussion points that were raised. All of this is, everything I'm skipping over is on the online, and I'm also happy to answer things in Slack or chat offline on video. And what I just want to end with is, again, this concept of the CD4 T cell as organizing the character of the response. So this is a slide of CD4 T cells that are differentiating into different types of CD4 T cells that will direct different types of responses. So responses that are characterized by distinct cytokines and distinct effector mechanisms. And so this response here, we often call a Th1 response. It's distinguished by interferon gamma, which is a cytokine, and it's distinguished by a lot of cell killing. So CD8 T cells are activated a lot in the response. This is the response you want when you have a virus. This is the response that everyone's looking for in SARS-CoV-2 papers, and it's what they found in people that clear the infection very quickly. And it's in contrast to another kind of response, the Th2 response, which is characterized by different cytokines, different transcription factors inside the CD4 T cell. And this is the response when you have a very large worm in your body and you need to get rid of the worm. You don't need cell killing because the worm is hundreds of thousands of times, sometimes bigger than your cells. So that's killing a couple cells on the worm, it's not going to do anything. So what you actually need is mechanical contraction of your colon, for instance, to expel a worm. And these cytokines are going to actually induce that response. And so the CD4 T cell has made the decision here that the infection is a worm, not a virus, and we're going to induce this kind of response. And so these are all critical concepts for the differentiation of these cell types. Okay. And then this slide I find really useful. During the introductions, you all were working on very different pathogens and thinking again about, for every pathogen, there's a different collection of effector mechanisms that are going to be useful for getting rid of that pathogen. That's what this slide tries to summarize up here, and hopefully my Zoom menu doesn't cover it up too much. Up here are the different effector mechanisms, at least for antibodies and cell-mediated responses, and down here are the pathogens, and then whether or not that effector mechanism is useful in controlling that particular pathogen. You see there's large areas of white where that effector mechanism doesn't really do anything to that pathogen. You can knock it out, the pathogen's cleared just as effectively, if not. And so there's some important specificity that comes from the actual effector mechanisms that are used. And then one last key point is that the immune system does have to shut itself off, and when it doesn't, you end up with autoimmunity and asthma and allergy. And in the online, I will talk about, in the online video, I talk about some of these mechanisms which are both cellular. So there are specific cells that help shut down the immune response called Treg cells. And these also have a number of different mechanisms, which are often immunological mechanisms to actually kill components of your immune response so that they stop doing what they are doing. And so I'm going to end there, back on this slide. And I'm happy to take questions again. I know we're over, but I think we did have a little bit of break time. And Andreas knows I always go over on the first day anyway, so. I am going to look at a couple of these questions that I didn't answer. Irene asked about the duration of memory, essentially, and I'm going to talk about that in the context of some of the specific pathogens that we are dealing with, particularly with SARS, because there's some really nice new data on that. Somebody asked about where dengue fits into the spectrum. Dengue, I classify as a classic kind of try to get in, try to get out viral replication, with the little twist that for dengue to get out of its host, it has to get bitten by, the host has to be bitten by a mosquito, but it really wants to have high viral titers so that when the mosquito bites it, it is transferred on. And so it's not, you can kind of think of it like a standard viral replication in the blood, just with this extra complication of the vector transmission. Questions about the 4 and 8 that I think I did answer. Somebody was asking about why this CDs are named the way they are. So CD stands for cluster of differentiation. And they, as Christopher says here, they were named randomly. They were named in the order that they were discovered, and there's virtually nothing useful you can use to try to remember them other than straight memorization, and it's unfortunate. I'm sorry. And the last question was, the role of IgA is fairly limited, but I don't think that was true. What's missing here in terms of the role IgA plays? So let's go back to that slide, probably covering it up again. So IgA was listed as important for influenza virus, polio, gonorrhea. It's a fair point that this might be underselling IgA. I will say the sort of definitive evidence that IgA is important is not really available. There's this assumption that IgA is really critical because, so IgA is an antibody that is secreted at mucosal surfaces. So your nose, your gut, your lungs are coated with IgA. And so there's a thought that to block these infections, you really want a lot of IgA. And you can kind of show that in some mouse models, but for a number of these different pathogens, I think the evidence isn't really there that IgA is super critical. And it's not to say that it's not critical, but going back to one of the questions that we had about redundancy, a problem with interpreting a lot of knockout data is even if you knock out IgA, which is doable, the other parts of the antibody responses, so IgG, IgE, these other forms of antibodies can take their place. And while it might not be ideal, it'll obscure the fact that IgA was important. And so your experiment will sort of give you this confusing result. And then the last question that just got posted is an IgA important for allergies? And the answer is no. You are thinking of IgE, which is another form of antibody that triggers histamine release from eosinophils and basophils, and so in mast cells. And so IgE is the antibody for allergy, and the actual probably purpose of IgE is actually against worms. So it is critical in the worm response.